Welcome to Young-Splaining Cellular Respiration and Fermentation. Cellular respiration and fermentation are catabolic processes. So it's a process that is going to take a compound and break it down into smaller compounds. When we're looking at respiration and fermentation, we're looking at the oxidation of organic fuels. Now, when we have an oxidation and reduction, oxidation simply means I'm going to take electrons from this compound. And a reduction is when that compound receives the electrons. So in cellular respiration and in fermentation, you're going to see redox reactions where something is being reduced and something else is being oxidized. The first step of the catabolic process is to identify whether or not oxygen is present. So if oxygen is present, which is what we see here, oxygen available to the cells, that process is going to be a complete consumption, so a complete breakdown of that organic fuel, and that is called aerobic respiration. Now, if there is no oxygen available to the cells, there's two possibilities. There will either be a partial consumption or a complete consumption of the organic fuels. If the organic fuels are only partially consumed, that's fermentation. However, if they are completely consumed, then we have anaerobic respiration. Notice that both fermentation and anaerobic respiration occur in the absence of oxygen, but only aerobic respiration occurs when there is oxygen. Another similarity to note is that both anaerobic and aerobic respiration is a complete consumption of the organic fuels. Now, looking further, cellular respiration can either be aerobic or anaerobic. Cellular respiration can consume carbs, lipids, or proteins. As a model, we look at what happens with carbs, and we use glucose as our model carbohydrate. Now, there are several steps to cellular respiration, and the purpose of this breakdown, this catabolic process happening in several steps, is to control the release of free energy. If it happened in one huge step, the energy change would be explosive. But by controlling that energy change, by making this happen in small steps, only a small amount of energy is released at each step. So let's look at the stages of cellular respiration. There's 3 stages and 2 locations within the cell. The important locations are going to be the cytosol, which is a dense fluid in the cell, and of course your mitochondria. There are 3 stages of cellular respiration, your first being glycolysis. Glycolysis will occur in the cytosol. It generates ATP through substrate level phosphorylation, and we'll also get some electrons taken to electron transport by using nicotinamide in its reduced form, what we'll call NADH. The second step is the citric acid cycle, but before we can move into the citric acid cycle, there has to be a pre-Krebs or a pre-citric acid process, and that process is going to take the pyruvate molecules created in glycolysis, and they're going to be oxidized through pyruvate oxidation to acetyl coenzyme A. Pyruvate holds the key to Krebs, but acetyl-CoA is the key to Krebs. So pyruvate has to be oxidized into acetyl coenzyme A, which starts the citric acid cycle, also known as the Krebs cycle. From the citric acid cycle, I'm going to have electrons carried through NAD, nicotinamide, which is going to be in the reduced form, and also FAD, flavin adenine dinucleotide, in its reduced state. Now, I'm going to make more ATP through substrate level phosphorylation as well. The last stage of respiration is going to be your oxidative phosphorylation. Oxidative phosphorylation has 2 parts, the electron transport and chemiosmosis. The ATP created through oxidative phosphorylation is an oxidative process, and 90% of your ATP is going to be generated through step 3, the oxidative phosphorylation, The goal of cellular respiration is to generate as much ATP as possible. Let's take a closer look at glycolysis. Again, glycolysis is going to occur in the cytoplasm. Therefore, oxygen is not a factor. In order for steps 2 and 3 of cellular respiration to occur in an aerobic process, oxygen needs to be present in the matrix of the mitochondria. But because glycolysis occurs in the cytoplasm, it is independent of oxygen. Glycolysis will occur in respiration, both aerobic and anaerobic, and in fermentation. Glycolysis means sugar split, glycosugar lysis split. And the sugar that we're going to look at is glucose. And you're going to have one molecule of glucose split into 2 molecules of pyruvate. Glucose is a 6-carbon compound. Pyruvate is a 3-carbon compound. So I'm going to take one 6-carbon compound and through a series of 10 steps, break it down into 2 3-carbon compounds. There's 2 phases of glycolysis. The first 5 steps is called the energy investment phase. During the energy investment phase, I'm going to use 2 ATP. The next 5 steps is called the energy payoff phase. During the energy payoff phase, I'm going to create 4 ATP, which gives me a net of 2, because remember I used 2, so it's like I have to repay those 2, then I still have 2 left over. So my net is 2. And I'm also going to produce 2 reduced nicotinamides, NADH. I'm going to end up with 2 nicotinamides and 2 pyruvates. So the net products of glycolysis would be your 2 pyruvates, and then I'm going to make 2 water molecules as a byproduct as well. I'm going to have 4 ATP formed. 2 of them were used to start the process, so I'm going to net 2 ATP. And then I am going to have 2 reduced molecules of nicotinamide, NADH, as well as 2 protons. Our next step is pyruvate oxidation. Pyruvate oxidation begins the breakdown of pyruvate. Pyruvate oxidation is going to be the process that begins stripping that organic molecule of its carbons and its oxygens. We don't need the carbons and oxygens, we need the hydrogens. Glucose is C6H12O6. We don't want any of those 6 carbons, we don't want any of those 6 oxygens, but we do want the hydrogens and the electrons that they carry with them. Pyruvate oxidation, again, holds the key to the citric acid cycle. When pyruvate is oxidized, it is going to reduce nicotinamide to NADH. Carbon dioxide will also be released at this process. Remember, pyruvate is a 3-carbon compound. When I oxidize pyruvate to acetyl coenzyme A, I am creating a 2-carbon compound. So, where the other carbon go? It's released in the form of carbon dioxide as a byproduct. Keep in mind, I have 2 pyruvate molecules, so this process will occur twice. So overall, I'm going to have 2 carbon dioxide molecules released and 2 nicotinamides reduced. Anytime nicotinamide or flavin-adenine dinucleotide are reduced, they are going to carry those electrons to the oxidative phosphorylation stage. They carry them to stage 3. Following pyruvate oxidation, it is important to know that pyruvate oxidation links glycolysis to the citric acid cycle. In order for pyruvate to be oxidized, oxygen must be present in the mitochondria. It begins the breakdown of pyruvate to carbon dioxide, and by converting pyruvate to acetyl coenzyme A, or acetyl-CoA. This is just a diagram showing the oxidation of pyruvate. So you start off with a pyruvate molecule. Here you have your 3-carbon compound. Carbon dioxide is removed. The compound is oxidized. That oxidation reduces nicotinamide. These reactions are going to be coupled. Your redox reactions will be coupled. One thing's being oxidized, something else is being reduced. Nicotinamide is going to be reduced to NADH. Coenzyme A will be added in. Now, coenzyme is just a cofactor. It's an enzyme helper, and it's called a coenzyme when it is organic. And what we end up with, the two of these carbons which were a part of pyruvate are still present. So we have this 3-carbon compound of pyruvate transform into a 2-carbon compound, an acetyl coenzyme A. And acetyl coenzyme A is the key to the Krebs cycle. Krebs cycle, also known as the citric acid cycle, occurs in the mitochondrial matrix. ATP, 1 molecule per turn, will be made of ATP. And we're also going to have some carrier molecules. You're going to make nicotinamide, and for the first time, you're going to make your flavin adenine dinucleotide. So you're going to make both NAD and FAD in this process. For each turn, you're going to get 1 ATP, 1 FADH2, and 3 NADH. Remember, there's 2 pyruvate molecules become acetyl coenzyme A. So per glucose, for every 1 glucose molecule that enters cellular respiration, aerobic cellular respiration, you're going to get 2 turns of the citric acid cycle. The last part is going to be, let's look at the citric acid cycle. So acetyl coenzyme A is the key that turns Krebs. It is going to bond with a substrate, which is a compound that is present in this cycle. We're going to get a release of your remaining carbons and oxygens. So you're going to get 2 carbon dioxide molecules out, and this is with both terms. You're going to get 3 nicotinamides reduced to NADH, produce 1 ATP through substrate level phosphorylation, and produce 1 FADH2. And remember, this is per turn. So overall, net 4 carbon dioxide release, 6 nicotinamides reduced, 2 ATP produced, and 2 FADH molecules. Now, to remember the steps of Krebs, I have a saying that says, "Please, Officer Amy, can I keep selling shoes for money?" The P in please represents the pyruvate molecule, which has to be oxidized. Officer Amy represents the oxaloacetate and the acetyl coenzyme A. In order for Krebs to start, acetyl coenzyme A has to bond with the oxaloacetate. CAN is for citrate. The first transformation happens when oxaloacetate bonds with acetyl coenzyme A. It creates what's known as citrate, which is why this cycle is often called the citric acid cycle. I is isocitrate. Isocitrate is an isomer of citrate, so citrate will be converted to its own isomer, isocitrate. Isocitrate is converted to alpha ketoglutarate, the K for keep. Between this step, the step that changes isocitrate to alpha ketoglutarate is going to strip the molecule of 1 carbon dioxide and also oxidize that molecule, reducing 1 NADH. The same thing happens with the next step. To go from alpha ketoglutarate to succinyl coenzyme A, carbon dioxide is removed, and NAD is reduced to NADH. At this point, I've gotten rid of all of the remaining carbons and oxygens that were a part of the original glucose molecule. Any remaining carbons are a part of the oxaloacetate, and I need to reform the oxaloacetate. Because without oxaloacetate, without Officer Amy, nothing can happen. So, our next step is between succinyl coenzyme A and succinate. This is where I'm going to get my one molecule of ATP formed. This is called a substrate level phosphorylation. Between succinate and fumarate, when succinate is transformed to fumarate, this is where flavin adenine dinucleotide, FAD, is going to be reduced to FADH2. A key difference between FAD and NAD, NAD carries 1 electron, FAD carries 2. You're going to see water put into the system between fumarate and malate. And then our very last step is when malate reforms oxaloacetate. And this is where that very last NAD molecule is going to be reduced to NADH. So your 3 NADs come in between isocitrate and alpha-ketoglutarate and succinyl-CoA, and between malate reforming to the oxaloacetate. Our last step of cellular respiration is oxidative phosphorylation. Oxidative phosphorylation occurs in the cristae of the mitochondria, which are the folds of the inner membrane. And the enzymes and proteins responsible for oxidative phosphorylation are actually embedded in this membrane. Chemiosmosis is going to couple electron transport to AT synthesis. Most energy extracted from food at this point can be found in your NAD and your FAD. So when you enter oxidative phosphorylation, the majority of the energy that was extracted from food is not yet found in ATP. It is still stored in the bonds between those hydrogens and those carrier molecules, NAD and FAD. NAD and FAD are going to donate those electrons to the electron transport chain, with oxygen serving as the final electron acceptor in aerobic respiration. If oxygen is not present in the matrix of the mitochondria, this process would stop with glycolysis. Pyruvate would not be oxidized, the citric acid cycle would not be turned on, NAD and FAD would not be bringing their electrons to the oxidative phosphorylation cascade. So this is a diagram showing oxidative phosphorylation. Oxidative phosphorylation has 2 parts, electron transport and chemiosmosis. Now what you see here, these purple proteins, some of them are transmembrane proteins. They actually go all the way through the membrane, and others are peripheral proteins. So you have protein complexes. There's 4 of them, 1, 2, 3, and 4. 1, 3, and 4 are all transmembrane, but 2 is going to be peripheral to the cytosolic side. Now, out here you also have what's known as a cytochrome, and that cytochrome is also a peripheral protein. These protein complexes are instrumental in electron transport. They alternate oxidation and reduction. So your carrier electron molecules, NAD and FAD, are going to bring those hydrogens to these protein complexes. NAD comes to 1, it carries 1 electron. FAD comes to 2, carries 2 electrons. That'll help you remember where they actually go. When NAD comes to this protein complex, the hydrogen is going to be basically dropped off and stripped of its electron. When that hydrogen molecule is stripped of its electron, the electron is going to be passed from protein complex 1 through a series of cytochromes to ultimate cytochrome Q, to protein complex 3, cytochrome C, protein complex 4, to the final electron acceptor, which is oxygen. Now, these cytochromes contain iron, just so you know, just a little extra information there. But when I strip a hydrogen of its electron, all I'm left with is a hydrogen ion, which we call a proton. Those protons are pumped out of the mitochondria into the space between the outer membrane and the inner membrane. So they're pumped out into the inner membrane space. This creates a charge gradient across the membrane. Now, the protons that are being pumped out are positive on this side, and I have my negative electrons being passed off to oxygen, making oxygen negatively charged on this side. So oxygen is being reduced. Now, when I have these oxygens carrying a negative charge and these protons carrying a positive charge, what do opposites do? They attract. So this is how we get water created as a byproduct, with the oxygen and electrons and those protons. But what about these guys that got pumped out here into this inner membrane space? Well, they don't want to be here. There's a difference in the concentration of hydrogen across the membrane. So these hydrogen ions, these protons, are going to come back in to the matrix of the mitochondria through a membrane transport protein. This membrane transport protein just so happens to be ATP synthase. And ATP synthase is the enzyme that catalyzes the reaction between ADP and inorganic phosphate to make ATP. The energy, the charge difference that is created as these protons re-enter the mitochondrial matrix is used to catalyze the reaction of ADP forming a bond with inorganic phosphates to make ATP. The formation of ATP using this proton gradient is called chemiosmosis. This gradient created is called a proton motive force because it is the motion of the protons, this force that's created by these protons, that drives the chemiosmosis. Your electron transport chain makes no ATP directly. What it does is it feeds those protons into the matrix and into the inner membrane space to create that proton gradient. It creates the proton motive force. Why is oxygen important? Oxygen is the final electron acceptor. Oxygen is very reactive. It is going to have a very strong pull on electrons. So when these electrons are brought to these protein complexes, oxygen is essentially sitting over here pulling those electrons down the electron chain. So they ultimately are accepted by the oxygen. Every time an electron passes from one reaction, one protein to the other one, there's a redox reaction going on. Redox reaction, something's being reduced, something's being oxidized, something's being reduced, something's being oxidized, all the way down. And that is an overview of cellular respiration. So, why are NAD and FAD important? NAD is a coenzyme. It's an oxidizing agent, meaning it is an electron acceptor. When it is reduced to NADH, it stores energy that's later tapped for ATP synthesis by passing electrons to the electron transport chain and adding protons to the proton gradient. So the oxidized form of nicotinamide is NAD+, and the reduced form is NADH. FAD is flavin adenine dinucleotide. It is also a coenzyme, but it is a double electron acceptor. It's an oxidizing agent, but it's a double electron acceptor. Same process as NAD. It's going to donate those electrons to electron transport, the protons to the proton gradient. Oxidize is FAD, just FAD. Reduce, FADH2. because it can carry 2 electrons. So that's your organic fuels being metabolized when oxygen is present. But what happens when there is no oxygen available to the cells? In the absence of oxygen, there's 2 processes possible: fermentation or anaerobic respiration. Now, animals are going to do aerobic respiration or fermentation, but there are some organisms that are capable to do anaerobic respiration. So what do these things have in common? Let's compare and contrast these two. First of all, they both occur without using oxygen. Because they're not oxygen dependent, they're going to get their ATP that is produced, that initial ATP comes from glycolysis. So they couple with glycolysis because glycolysis does not depend on the presence of oxygen. Now, fermentation is going to be a partial consumption of the organic fuels. And it does use substrate level phosphorylation. It does not use electron transport to generate ATP. So there is no oxidative phosphorylation, but there is substrate level phosphorylation. And this substrate level phosphorylation happens in glycolysis. The purpose of fermentation is to regenerate nicotinamide in its oxidized form. Nicotinamide does not need to be bound to hydrogen indefinitely. Remember, during the second 5 steps of glycolysis, one of the products is the reduced form of nicotinamide. Well, it can't stay that way if it cannot carry those electrons to electron transport. If there's no oxygen, it cannot carry them to electron transport. So it has to be regenerated. It needs to be oxidized again in its oxidized form. The final electron acceptors in fermentation is either going to be pyruvate or acetaldehyde. And there's 2 types of fermentation: common fermentation, alcoholic fermentation, and lactic acid fermentation. Now, anaerobic respiration is going to be a complete consumption. It's going to use the electron transport chain, unlike fermentation, which does not. The only difference between anaerobic respiration, well, the main difference between anaerobic respiration and aerobic is the final electron acceptor is not oxygen. It's usually something like sulfate, but it is not oxygen. When you look at your 2 types of fermentation, you have alcoholic fermentation and and lactic acid fermentation. In alcoholic fermentation, pyruvate is converted to ethanol in 2 steps. Alcoholic fermentation also breaks down pyruvate a step further by getting rid of carbon dioxide. So you end up with 2 ethanols and 2 carbon dioxide. Lactic acid fermentation's job is only to reduce nicotinamide to NADH. And by doing so, you end up with 2 molecules of lactic acid, often called lactate. There is no carbon dioxide released in lactic acid fermentation. So again, here's an overview. Glucose enters through glycolysis, is broken down in pyruvate in the cytosol. If no oxygen is present in the animal cell, it's going to do fermentation. Fermentation can be either alcoholic or lactic acid. If oxygen is present, pyruvate is oxidized to acetyl coenzyme A. That acetyl coenzyme A will start the Krebs cycle, and the end result from the Krebs cycle will be nicotinamide and flavin adenine dinucleotide being reduced and carrying those electrons to electron transport for oxidative phosphorylation and driving chemiosmosis. I hope this helps. My next step is to go through each one of these processes step by step. Watch video 2.